Good evening, everyone. We'll wait for a few more minutes to let others join, and then we can start in another 5 minutes. Good evening, everyone. Let me just share my screen. Can you all see the presentation?
Yes, ma'am.
Yeah, thank you. So, hello everyone, and welcome back to week 11 of the PMRF TA sessions for NPTEL online course Drug Delivery Principles and Engineering. And for nearly 10 weeks, we have been discussing about different concepts of drug delivery, tissue engineering, and so on and so forth. And let's look at some of the key points from this week's lectures. So in the last week, we have seen about gene therapy. We learned about what gene therapy is, and first of all, why it is done. Because sometimes due to some mutations in the genes, first of all, genes are a part of your DNA which actually code for the proteins that runs the body. And sometimes what happens, because of mutations, there might be that some of the genes required for an important protein is missing from a person's body, or it gives rise to a faulty protein which is not functional. So in that case, to treat such a particular, so giving medicines will only will not attack at the root cause of this particular disease. So in that case, what you can do, you can use gene therapy. You can deliver a gene which was missing from the patient's body, or you can deliver the correct gene which will then eliminate the faulty gene and give and produce a protein which is actually functional in the right way. So this is what gene therapy is used for. So we saw that to one approach to gene therapy is you get a patient, you know what gene is missing in the patient's body. So what you can do, you can collect cells from the patient's body. You can do genetic engineering to these cells. So ex vivo, you can deliver the gene inside the cells, and then you put these cells back into the patient's body, which will then the gene will then be transcribed and translated into the correct protein, and in that way you can try treating the particular disease. But through literature, we have seen that this approach is not very successful, although a lot of research has been going on in this approach. Just a minute. Yeah, so an alternative to that is, what if instead of taking out the cells and then giving the gene, you can just deliver the gene to the patient's body, right? So you take the gene and you can just inject it into the patient's body. And then we also saw that there are several, although this approach sounds very nice, there are several barriers to giving gene delivering gene as such. So these could be, one is that when your gene is inside the blood, there are a lot of, since you'll be injecting it, it will be flowing inside the blood. So a lot of enzymes are present in the blood serum, which would then degrade your gene, and the gene will not even reach the cells. Next barrier could be, so there are several barriers to this. Next barrier could be your permeability in the cell. So we all of us know that cell membrane is negatively charged, and so is your DNA. So the DNA is also negatively charged. So the, so it will the charged molecules will not be taken up immediately by the cell, right? So it will have to go through some channel. So there is another barrier. Even if the gene reaches the cell, it will not be taken up very easily. Then let's say somehow your gene overcomes all of these barriers and actually reaches the cell. It will be taken up by endocytosis, but your cells are very uh, tuned to this. They're trained to take things inside the endosome and lysosomal compartment, wherein the pH is very low, and this is done to degrade any extracellular debris that could be there. So because of this harsh environment, it could also degrade your gene, and then your gene will never, so for your gene to be functional, it has to reach the nuclear membrane, it has to reach the nucleus for it to be transcribed. So, so if it doesn't reach the nucleus, then the therapy is again a fail. So several such barriers are there, and so there are ways to overcome these barriers as well. So now that you saw that uh, your gene first of all has to survive the serum enzymes, and it has to enter the cell. It from the cell, it has to come into the cytoplasm. From the cytoplasm, it has to go to the nucleus. So all of these barriers could be overcome by using something known as vectors. So there are these vectors could be of two types. You can use viral based vectors, or it could be non-viral based vectors. So what are these vectors actually? It's nothing but you use a carrier to take your gene from your solution to in such a way that it will pass all these barriers, and it will then enter the nucleus. So let's look at one of these. This is the viral based vectors. So what you can do is, so viruses, we all know, are trained through a lot of evolution to infect particular cells. So if in case you want to deliver a gene to a human, what you can do, you can take human viruses which infect humans, which are very good at infecting human cells. You can take these viruses, you can remove the part that causes the disease in the humans. So you take the viral genome, and you delete some of the genes in the viral genome, which would then lead to disease. So after deleting these genes, you can put your gene, and then let the virus infect the cells in the human patient. So what this will do is, your virus already knows how to overcome all of these barriers. It has a very high transfection efficiency. It knows where to go, which cells to target, how to evade these endolysosomal enzymes, how to protect its own genome from getting degraded. So what these viral vectors will do, this is a live virus. First of all, it's able to infect the cells, and then what it will do, it will inject the its own viral genome into the cell, and then that genome will then be integrated into the genome of the cell, the human cell that the particular patient cells, and then that will be transcribed and translated, thinking that the virus will think that it is its own genome. So it will not cause any disease because you have deleted the genes which are responsible for causing the disease. But then you have put your own gene there, whichever you want, and that will then lead to the protein which was missing from the patient's body. So this is one approach for doing gene therapy. But as with every other system that we have seen throughout the course, it will have its own advantages as well as disadvantages. So the first advantage is it has a very high transfection efficiency. Transfection efficiency meaning it is very good at infecting its the transfer of the gene from the virus to the mammalian cell is very good. It is cell specific. It knows which cells to target, so it will eliminate the off target. It will be very specific, meaning that it will not go to other cells and infect those cells. Then it has evolved mechanisms for endosomal escape. So in that case, your most of your gene material will not be degraded, and it will reach the particular site. Talking about the disadvantage, first of all, it will have a very strong immune response. Now, even though you have deleted the genes which are responsible for causing the disease, but the virus is still pretty much alive, and it's a foreign material for the patient's body, for the human body. So your immunity is going to be very strong against this virus, so it will try eliminating the virus. Next disadvantage could be it might activate oncogenes. So it's a very good thing that the virus is able to infect the gene, and it integrates that gene in the genome of the cell, but then it does not have a control of where that gene is going to be integrated. It might be that it integrates in such a way that it might trigger some of the oncogenes. What are oncogenes? Oncogenes are responsible for causing cancer. So if that happens, then you have unintentionally triggered an oncogene in the patient's body. So instead of treating one particular disease, you have now made the problem even worse. So this is one of the disadvantages. This could happen. And also there is a limited size of the gene. So the virus will have its own genome. It has some limitations on its own genome size, because it has to pack all this genome into its viral particles. So there's not much space or not much scope of putting in larger genes. And it could also lead to viral disease. Now, even though I am saying that you have deleted the genes responsible for causing the disease, but then in some cases it might happen that the virus, the deletion has not happened in a very proper way, and it could lead to viral disease. So these are some of the advantages and disadvantages of using viral vectors. So to overcome some of these advantages, what we can do, we can use non-viral based vectors. So these are your polymer and lipid based vectors. What is this? This is nothing but you take a polymer, you encapsulate your gene in that, and you deliver these particles, these polymer particles. So these could be your micro or nanoparticles. So it comes with a lot of advantages. First of all, it has a lower risk of immune response, because so there are a lot of polymers which are biocompatible, so they will not elicit a very strong immune response once you deliver it into the patient's body. Since these are foreign, some amount of immune response would be there, but then this is controllable. This won't hamper the body much. Then there is no gene size limitation. You can put as large a gene as possible after optimizing what polymer to use and what size of particles to make. No integration in genome. So since these are not virus particles, your particles will just, carrying the gene will just go to the cell and they will stay in the cytoplasm, or the gene will just go to the nucleus. It will not integrate into the genome, so there is no chance of activating oncogenes. Now, talking about disadvantage of this non-viral vector-based method is low transfection efficiency, because in case of virus, it was naturally evolved to infect mammalian cells, but in this case, polymers don't know how to infect, and so the transfection is very less, meaning your whatever gene you have encapsulated, not all of it will be getting into the cell. So there's low transfection efficiency. It could be toxic at higher doses, because now the transfection efficiency is low. To achieve the therapeutic effect, you will try delivering more and more particles to the patient's body. Now, even if the polymers are biocompatible, above a certain concentration, they are definitely going to turn toxic. So we need to be careful about how much polymer we are delivering into a patient's body. And also, they have very poor endosomal escape, because as I said, these particles will be endocytosed into the patient's cells, but then they will end up into the endosomal compartment. So there the pH is again very low, which could then degrade your particles, and in that process will also degrade your DNA, the gene that you have delivered. So these are some of the disadvantages. And talking about this endosomal escape, what we can do is we can use cationic polymers. So we are still on the theme of using polymers. So since this is a drug delivery course, we'll focus more on the non-viral vector-based methods. So what kind of polymers you can use for delivering genes? They could be cationic polymers, meaning they're positively charged. Now, first of all, why positively charged? Because as I said, your DNA is negatively charged. So to have a very good encapsulation efficiency, we will take something that is positively charged. So they will make a very nice complex there along with your DNA. So more amount of DNA could be loaded onto these particles, which would then be easier for delivery. So some of the examples of these cationic polymers are PEI, which is polyethylene imine. Then there is chitosan, there is dendrimers, so etc. There are a lot of positively charged polymers which are available in the market to do research on this gene therapy. So the next point that was discussed in this week's lecture was using DNA as a vaccine. So a few lectures back, you might remember that we started talking about vaccines, right? So what are vaccines? Vaccines are basically part of a pathogen, or the entire pathogen itself, against which you want the patient's body to be immune. So these are some of these could be proteins. So taking some of the proteins, you first deliver them in a very small amount to a healthy individual, and then that individual's immunity will be triggered against these proteins, and the memory cells will remember these proteins. So in case when there is an actual infection, the body's immunity already remembers, because of these memory cells, that this is the particular protein we are supposed to fight against. Then it will launch a very strong immune response, which will then eliminate that particular disease. So instead of delivering proteins, what you can do is you can deliver the DNA, which then codes for these proteins as a vaccine. So that's one of the ways of using DNA as a vaccine. So the most important advantage of using DNA as a vaccine is because it can trigger both humoral, meaning your antibody based immunity. It can trigger both humoral as well as cellular immunity. So it can trigger both B cells as well as T cell immunity, which is a very good advantage. Then the next point that was discussed was cancer vaccine. So in the last week also we got introduced to this topic known as cancer vaccine. So these could be your prophylactic or therapeutic vaccines. How can a cancer vaccine be prophylactic? So for example, there are some of the viruses which are known to cause cancer in humans. So if you know that in that case, what you can do, you know the virus, which virus is going to cause that particular cancer. What you can do, you can make vaccine against these viruses. So if you deliver these vaccines to the humans, when there is an actual infection from the virus, your body is already ready to fight against this particular virus. So in this way, prophylactic vaccines could be manufactured for cancer. And then we have therapeutic vaccines. So how does this work? So in case you get a patient who is already suffering from a particular type of cancer, in that case, the patient's body may not be that strong. The immunity in that patient's body is not that strong to fight against this cancer. So the goal of therapeutic cancer vaccines is to strengthen this immunity against these particular antigens which would have been occurred because of the particular type of cancer. So, and then this therapeutic vaccines could be manufactured in two different types. They could be autologous tumor cell vaccine. What does this mean? You get a patient. Let's say that patient is suffering from a lung cancer, and you want to give cancer vaccine for this particular patient. So what you can do is you can take some of the lung cells. So these cells would be mutated, right? They have cancer. These are tumor cells. So they will have some antigens, some proteins which are changed from the normal protein, and then you can engineer as an antigen. So this will be an antigen for the patient's body. So you can take the tumor cells, you can chop them up. So this will give you a nice cocktail of the proteins which are being mutated in the patient's body. And then you can deliver these proteins as a kind of vaccine along with some adjuvant. You must remember that adjuvants are helpers in strengthening the immune system. So in this way, you can then manufacture an autologous tumor cell vaccine. Or on the other hand, what you can do, so in case you get a patient who is having lung cancer, you can go back to your records and see how many patients. So there would be patients who had come earlier to the clinic who had lung cancer. So what you can do, you could have taken cells from them and developed a cell line, and then use the antigens from these particular cell lines to then give to the new patient. So in this case, it will be an allogenic tumor cell vaccine. Why allogenic? Because it's not taken from the patient's own body. These are taken from a different body which is from the same species. So both of these prophylactic as well as therapeutic vaccines are in research. Mostly the therapeutic cancer vaccines are in research phase. So these are some of the points that were discussed in this week's lecture. So we can then look at some of the assignment questions. If I'm going too fast, you guys can let me know and I can slow down a little. So the first question is, select the correct option with regard to vaccines for cancer. We have 5 different options, so you can take your time in reading the options and let me know if anybody knows the answer.
Ma'am, option 2 is correct.
Okay, these vaccines are agents that can recognize and attack tumor cells. Okay, anybody else? Thanks for answering. Okay, someone is saying option 3 in the chat box, which is by isolating cells from autologous tumor tissue, a wide range of antigens can be presented. Okay, thanks for answering. So we have 2 different responses. Anybody else has a say on this? No?
Option 2.
Okay, we have one more response saying option 2. Okay, thanks for answering all of you. The correct answer is actually option 3. So if you just read about vaccines for cancer, we'll just go through the options one by one. They have to be therapeutic, as prophylactic cancer vaccines are unsafe and cause tumors, which is not correct. We have seen a few examples of prophylactic cancers which could be given for cancers caused by viruses. Second option was, these vaccines are agents that can recognize and attack tumor cells. No, that's not actually correct. These vaccines are not going to attack the tumor cell. These vaccines are going to train your immune system to attack those particular tumor cells. So the vaccines are not directly responsible for attacking tumor cells. By isolating cells from autologous tumor tissue, a wide range of antigens can be presented. This is one of the most important advantage of using autologous tumor tissue. So this is correct. Adjuvants are not required for tumor vaccines. That is false. You need adjuvants. You need the immune system to know that this particular antigen is actually causing tumors. So adjuvants are used to strengthen the immune system against whenever you use it for tumor vaccines. Cancer vaccines are used for training neutrophils to attack the cancer tissue, thereby annihilating them. No, vaccines are actually used for training your B and T cells. So if you read through this slide also, you'll get to know the correct answer. But yeah, in this case, the correct answer is option 3. Is this clear?
Yes, ma'am.
Okay, thank you. So let's move on towards the next question. Next is, therapeutic cancer vaccines are a type of active what with the aim to produce a blank immune response to tumor antigens or tumor associated antigens that are able to blank cancer cells. So you just have to fill in the blanks. and with 4 options, and let me know if anybody knows the answer.
Option 2.
Okay, option 2, immunotherapy, specific and destroy. Okay, anybody else? Thank you for answering.
Option 3.
Option 3 is immunotherapy, broad and destroy. Okay, any other?
Option 2.
Option 2. Okay, so we have 2 responses with option 2 and 1 with option 3. Others, do you have any answer? Okay, no one. Let's look at the answer. Yeah, the correct answer is option 2. So let's fill in the blanks and read the statement again. Therapeutic cancer vaccines are a type of active immunotherapy, that is correct, with the aim to produce a specific immune response to tumor antigen. So this is a specific immune response. It's going to trigger only against the specific antigens or tumor associated antigens that are able to destroy cancer cells. So all the things we are doing is because we want the therapeutic, we want the immunity to be very specific against the tumor antigens, and we want these tumors, the immune cells, to then destroy these cancer cells. Is it clear?
Yes, ma'am.
Thank you. Okay, let's move on towards the next question then. Next is, what kind of polymers are generally used in gene delivery? We have 2 correct options to choose from. I think we just discussed this in the... Yeah.
Cation and positively charged.
Yeah. I think 2 people answered. So yeah, in such questions, please be careful whenever it says you have to choose 2 correct options. So you're correct. It is the polymers that are used in gene delivery should be cationic and positively charged. So that it's first of all easier for them to complex against the negatively charged DNA, and then this also helps in the endosomal escape. Yeah, you can read this in the slide as well. The 4th question is, what makes chitosan a suitable polymer as a gene carrier? I think it's now getting repeated. So yeah, again 2 options to choose from.
Option 1 and 2.
Okay, thanks for answering. Anybody else?
Option 1 and 2.
Okay, yeah, thank you both of you. So yeah, both of you are correct. The correct answer is positively charged and biocompatible. So your chitosan is used mostly as a gene carrier because first of all it's positively charged and it's biocompatible. So you have to choose a polymer which is biocompatible, because then you don't want the immune response to be elicited against the polymer itself. So yeah, that's all in this question. So the next question is, which of the following polymers are not commonly used for gene delivery?
Option 1 and 2.
No, you have to choose only one option.
Option 1.
Okay, thanks for answering. Anybody else?
Option 5.
Option 5. Okay, two different responses we have got until now. Anybody else has a say in this?
Sorry, ma'am. It's option 2.
Okay, so you're changing your answer from 1 to 2. Okay, no one else? Okay, let's look at the answer. The correct answer is PVC. So the question is asking us what is not commonly used for which polymer is not commonly used for gene delivery. So if you see all of the first four options, these are all positively charged polymers. Only PVC is a negatively charged polymer, hence it's not commonly used for gene delivery. Clear? I'll move on towards the next question then. Next is, which of the following is false for allogenic tumor cell vaccines? Okay, sorry if I'm missing the answers posted in the chat box. Any volunteers for this question?
Option 1.
Option 1 is uses few established human tumor cell lines. Okay. Anybody else?
Option 4.
1, 2, 3, 4. Risk of autoimmunity. Okay. Thanks for responding, both of you. Anyone else wants to give it a try?
Option 4.
Option 4 is again risk of autoimmunity. Okay. Okay, let's look at the answer then. So the correct answer is highly successful approach. So please read the questions carefully. It's asking us what is false about allogenic tumor cell vaccines. So what are allogenic tumor cell vaccines, which is discussed in the introductory slide. So these are taken from few established human tumor cell lines. So this is actually true, the first option. Second is it has a limitless source and it's cost effective. Why? Because these are cultured ex vivo. These are cultured in labs. So you don't have to always take it from any patient, any live human. So that's why it has a limitless source. You can keep on culturing these cells. That makes it cost effective. Fourth is risk of autoimmunity. That won't happen in allogenic tumor cells. So autoimmunity happens uh, when your immune system is targeting your own cells. So this risk happens in the autologous tumor cell vaccines, and it may not be effective at all, which is actually true. Why? Because these tumor cell lines are developed from some other patient, right? It's not necessarily that your patient will have the same kind of antigen, same kind of protein mutated as any other person. So it might not be effective at all. The only false option here is it's a highly successful approach, which actually it is not for allogenic tumor cell vaccines, because it might not be effective at all. So that's why this is not a very highly successful approach. So this option 3 makes it false for allogenic tumor cell vaccines. Is this clear?
Yes, ma'am.
Okay, thank you. So let's look at the next question. The next is, which of these is not a potential tumor immune evasion mechanism? We have a lot of options to choose from. Only one option is correct. So your tumor cells in the body are they're very smart. They know how to evade the immune mechanism. So yeah, which of these is not one such mechanism?
Ma'am, last option.
Last is upregulation of antigen processing. Okay, thank you for answering. Anyone else? Okay, no one. Yeah, you're perfectly correct. The upregulation of antigen processing is not a potential tumor evasion mechanism. So you can read it in this slide also. So basically what this question means is, whenever a tumor occurs in your body, your immune system is very trained to eliminate these particular tumor cells. So it routinely keeps on doing this. So because the DNA replication is not a very foolproof method, that it might happen that some mutations keep on arising in our cells whenever they are replicating. But the immune system is very good in identifying these particular changed mutations, the changed antigens, and they take care of these tumor cells. But sometimes it might happen that the mutation is a very small mutation. Let's say only one amino acid is getting changed in that particular protein. So it becomes difficult for the immune system to identify these tumor cells. So in that case, the tumor cell will not be eliminated and it will establish itself, and then it can then have different other mechanisms to avoid this immune system. So these could be, it has physical barriers imposed by tumor architecture. The tumor which then grows has a very tight architecture. It will not let your immune systems come nearby it. And then you have induction of host myeloid and lymphoid suppressor cells. So it will actually suppress the immune cells, which is one of the mechanisms. Then it can also block T cell effector function. So T cells are required to eliminate these tumor cells. So they can block this function. They resist apoptosis. They downregulate the MHCs. So MHCs are required for antigen presentation. which then helps your T cells to identify that these particular cells need to be killed. Then evasion of T cell recognition, that is also one mechanism. The only mechanism that does not fit in this particular tumor cell evasion is upregulation of antigen processing, which is false. It actually downregulates antigen processing. That's how it escapes the immune system. So I hope I'm making sense with this explanation. Okay, clear? Fine, then I'll go to the next question. The next is, what are not the objectives of gene therapy? So we have to select two correct options.
Option 5 and 6.
Option 5, okay, the last two options. Okay, thanks for answering. Anybody? Anybody else? No other response. Let's look at the answer then. Yeah, you're correct. The last two options are not the objectives of gene therapy. Replacing a missing or defective gene, yes, that's what gene therapy is done for. Repair a non-functional gene, produce therapeutic proteins that help treat a disease, or vaccinate a person against an infection. These are all the objectives of gene therapy. Cleave healthy genes already present to generate haploid cells, cause dysfunction at a cellular level. This is what we don't want. So these are not the objectives of gene therapy. Pretty straightforward, I feel. Yeah. Okay, let's look at the next question. Next is, what property of the polymer causes proton sponge effect?
Presence of secondary and tertiary amines.
Yeah, you're correct. The correct answer is presence of secondary and tertiary amines. So we saw this proton sponge effect in the initial lectures, right? So what this is basically, whenever your particles are uptaken by the cells through endocytosis, as I said, they'll land up in the endosome and lysosomal membranes, where the pH is very, very low, somewhere around 5. So, and that's where they'll be degraded. So what we have to do is, we don't want our particles to be degraded there. We want the particles to escape that particular endosome and land up in the cytoplasm. Now to do that, what we can do, we can use polymers which have a lot of secondary and tertiary amines, which will then take up all the, they are able to, because of the presence of these amines, they are able to sequester, they are able to absorb all the H plus that are present in the H plus ions that are present in the compartment. If that happens, then the cell thinks that something is going wrong and it has to maintain the pH of the endosome. In that process, it will pump most of the H plus ions from the cytoplasm to the endosomal compartment. Now, because of a lot of H plus are getting pumped into the endosome, it then creates imbalance. So to balance that, it will also pump in chloride ions to manage the ionic strength there. And in that process, now there is a lot of ions present in that endosomal membrane, endosomal compartment. So this creates an osmotic imbalance. So to balance that, a lot of water content will be pumped, will be taken in the endosomal compartment, and all of this now creates a pressure on this endosomal membrane, which will then burst after a while. So once it bursts, all the contents of the endosome will be leaked into the cytoplasm. So in that process, your particles will also enter the cytoplasm. So this is known as proton sponge effect, and to do that, we need polymers which have a lot of secondary and tertiary amines. Okay, is this clear? You also have the explanation in this particular slide and in some of the initial presentations. Okay, then let's move on towards the last question for this assignment. So, which of the following can be considered as an extracellular barrier for gene delivery? Again, we have to choose two correct options. Anyone wants to volunteer?
Option 3.
Okay. Option 3, that is degradation of DNA in plasma. Okay. You have one response. Since we have to choose two correct options, you can also guess at the second option.
Option 1 and option 3.
Okay, yeah, you're right. The correct answers for this question are option 1 and 3. So, as I said, there are a lot of barriers for gene delivery, right? So to better understand these barriers, they are divided into extracellular and intracellular barriers. So your extracellular barriers are inhibition of transcription by mucus, because there is a lot of mucus lining all the your body. So your tissue will also have extracellular matrix as well as mucus. So this will not let your DNA enter into the cell. And the next is degradation of DNA in plasma, which we already know because of the serum enzymes. This could be degraded. All the rest of the options are also barriers for gene delivery, but these are mostly intracellular barriers, like uptake of DNA by mitochondria, lysosomal degradation. This all is going to happen once your gene enters the particular cell. So I hope this is clear, and this was the last question for the assignment. So if anybody has a doubt in all the rest of the questions or wants me to repeat a particular question, we can go through it. If no, that's all I have for you for this class, and then we can meet again in the next week. So that would be our last session, week 12. After that, I'd also be having an extra session to discuss some of the numericals. That would be next Monday. Yeah, does anybody else have a doubt in today's session? Or regarding any of the other concepts which you've gone through? We can discuss that now. If not, then we can stop the meeting here, and I'll meet you people next week. Is that all? Shall I close the meeting then?
Yes, ma'am.
Okay, sure. Thank you. Thanks for joining today. Meet you people next week.